Okay, let's look at mass flow hypothesis in phloem. So as you can see over here, I don't wish to elaborate on each individual parts like xylem, phloem and so on. Okay, so let's start with the first part of the mass flow of hypothesis, which is sucrose loading. As you all know that the leaf cell, which acts as a source, undergoes photosynthesis and produces sucrose, the food for the plants. So what happened actually, the companion cells over here contains specific protein pumps over here, the region that I'm showing here. Okay, something like this. Yeah, let me enlarge it so that you can see. Okay, yeah. So we have two protein pumps over here. The first one is called proton pump, or also known as H positive ATPase. Okay, the main function of this proton pump is to remove the hydrogen ion from the companion cell into the source. Yeah, so from the companion cell into the source. So as you can see over here, it spends ATP, which means this process requires ATP. Okay, so when this process requires ATP, the hydrogen ion is pumped against the concentration gradient into the source. So what happens over here, the hydrogen ion together with the sucrose molecule is co-transported together back into the companion cell. So as you can see over here, the H and the sucrose are co-transported back into the companion cell. This is how a lot of sucrose is sent to the companion cells over here. Once the sucrose is already inside the companion cell, the sucrose can just diffuse into the phloem by using this plasmodesmata. It's like a small opening over here. Okay, as you can see over here, there are a lot of sucrose over here, and this part of phloem is called sieve tube. And now we can move to the second part of our translocation, which is the mass flow part. Okay, when the sucrose is inside the sieve tube, what happens? It reduces the water potential level. When it reduces the water potential level, so generally the water from the xylem follows the concentration gradient into the sieve tube. So what happens over here? The water dissolves the sucrose and creates high hydrostatic pressure at the top over here. And when you have high hydrostatic pressure over here, and as you can see, generally here when the water moves out, you will have low hydrostatic pressure. The sucrose will move from the high hydrostatic pressure area to the low hydrostatic pressure area. This is what we call as mass flow. And let's look at our last part for today, which is the sink loading. So following the mass flow from the high hydrostatic pressure to the low hydrostatic pressure, once the sucrose is already bottom over here, it just diffuses into the companion cell and diffuses into the sink area. Okay, there's very important process that happens over here. Over here in the sink, the sucrose converts itself into starch. Why? Because starch does not affect the water potential inside your sieve tube, so that the mass flow over here can continue to occur. So that's it for today. Let's look at other part next time.